Hello, I'm Phillip Morin from the Southwest Fisheries Science Center in La Jolla, California. I got started working on cetacean genomes a number of years ago, and that's how we came about with the Cetacean Genomes Project. My first genome analyses were of the critically endangered vaquita porpoise, and we generated the genome of the vaquita because very little was known about this species, and that's true of many cetaceans, whales, dolphins, and porpoises, because we cannot go out and collect a lot of field information on their evolutionary history, ecology, taxonomy, and demographics. So we use the vaquita genome as a way to understand something about the species, to be able to figure out whether we could apply that towards conservation. When we sequenced the vaquita reference quality genome, the first thing we found was that it had about the lowest genome-wide diversity or heterozygosity of any mammal species to date. This was uniformly low across the genome. And the important thing about this was that we could use that to infer the demographic history, and learned that this low diversity was due to a very long time of being a small population. And by long time, I mean hundreds of thousands of years, where the vaquita had persisted in its environment as a small population. The critical information from that was that allowed us to also look at more individuals and determine whether that low diversity was due to the recent changes in the population or something historical, and how that affected the mutational load or inbreeding depression probability of species that is currently very small. That is a number of less than a dozen individuals remaining. So we used the genetic information, the genomic information, to then incorporate into predictive models of population survival and recovery under different scenarios. And indeed, we found that this low diversity was actually a good thing in this case for the vaquita to be able to survive, reproduce, and recover if we're able to reduce the bycatch, which is killing the species currently. And this was all published recently and has shown us the power of genomics for doing conservation planning. So this was where we got into understanding that genomics could really be useful for a lot of cetacean studies. And our goal, was to generate international collaboration that would help collect and preserve samples that were necessary and appropriate for reference quality genome sequencing, and then help share and coordinate that information among researchers and field workers who could then move forward with generating genomic data with the avoiding redundancy, reducing costs, and also seeking funding to help targeted species to fill in the gaps across the cetacean phylogeny. So on the right here, you see the phylogeny of about 93 recognized species of cetaceans. And as of 2020, when we were starting this collaboration, there were 3 species that were either completed or nearly completed to the point of a reference quality genome. These were all in collaboration with the Vertebrate Genomes Project. And our goal was to try to expand that, to get as many reference quality genomes across the phylogeny as possible, ultimately to get them all, and similar to the Darwin Tree of Life and the Vertebrate Genomes Project goals. To do that, we needed to coordinate information, like I said, so we set up a website that collects all the information, kind of a storehouse of information about samples and genome efforts, and then to move forward with targeted sequencing. So as of this year, 2022, we now have, we've made a lot of progress. We have 11 species either completed or in progress, and these were targeted to be distributed as much as possible across the phylogeny in the major families of Cetacea. Of course, this creates what I call the infrastructure, the kind of having those target species within a group of closely related species to use as a genome resource. But ultimately, what we want to do is fill out the whole tree with as many genome resources as possible. So as of now, The green bars in this chart show the ones that I just mentioned, the 11 reference quality genomes that are in progress or nearly complete. There's another parallel project the DNA Zoo is working on in the US to generate draft genomes. These are high-C scaffolded genomes for 25 additional species for which samples were available in the US a number of years ago when this project was planned. So that brings us up to a good number, over 36 species, about a third of the cetacean species, where we have some either a draft or reference quality genome assembly. But there are 57 species at the time we were planning this that did not have any plans or identified samples moving forward. So in the last couple of years, we've been working on identifying those samples. First of all, the high quality samples that could be used for reference quality genome sequencing, and specifically working with the Darwin Tree of Life and the UK Stranding Network, as well as the US National Marine Mammal Tissue Bank, to identify species in the North Atlantic that were candidates for sequencing by the Darwin Tree of Life, and for which we could identify a high quality tissue sample to be used for DNA and RNA extraction. So we've identified about 26 of those that are now planned, hopefully moving forward in the next year. And then for many species, we know we can't get those high quality samples. So we're working on identifying lower quality samples that cover the diversity of species. And specifically, the Southwest Fisheries Science Center and DNA Zoo have been working together to identify lower quality skin biopsy samples for an additional 34 species that we're now currently triaging those samples to identify which ones we can send to the DNA Zoo for high C and whole genome shotgun sequencing for scaffolded draft genomes. So that gets us up to almost all of the species that we have some genome project planned in the next year or so to get started on that. I've mentioned multiple times that tissues are the critical bottleneck for cetaceans because they are so hard to collect samples from, and especially to get those high quality samples that have to be preserved immediately in particular ways. So we've worked with the San Diego Zoo for the last more than 15 years to opportunistically identify samples that can be collected appropriately and sent to them for tissue culture. And we now have 17 species there that have, some of which have been used for these sequencing projects I've been talking about. And also, like I said before, the stranding networks in the UK and the US, they go out and identify animals that strand on the beach, and if they're fresh enough, they can collect tissues. And we're working to help them both identify tissues that have been preserved in the past, as well as samples that they can collect in the future and preserve appropriately and funnel into our sequencing projects. And with that, I will leave you with the website address there and my email address, and would like to recognize all of the people who are involved in collecting samples, preserving those samples, the databases that help us understand the quality of those samples, where they're distributed, and all the people that help to do the sequencing and assembly of these genome projects. Thank you.